Good afternoon, everyone. My name is Sofia Teran, and welcome to another talk of the Phylogeography Symposium. Thank you for assisting to this symposium. And today I'd like to introduce to you Dr. Diego Alvarado-Serrano, who is going to talk about Akodon mollis today. He is part of the Department of Biological Sciences of the Ohio University. So please, Doctor, start presenting.
Thank you. Let me just set this up. So hopefully all you can see my slides now. So first, thank you for your interest in this talk and to the organizers for inviting me to present some of the work I do. I'm really excited of everything that I've heard so far on this symposium, and I'm looking forward to interacting with some of you later on. So, as I said, I'm going to talk about the use of integrative phylogeography to study evolutionary consequences of climate-induced distributional shifts. But before doing that, I want to sort of orient you as to where this talk fits within the context of phylogeography. So phylogeography was introduced as this field that connects both population genetics and phylogenetics. And originally, the main goal was to describe patterns of molecular variation across geography, and using these patterns, make inferences about the evolutionary processes that lead to them. However, it was soon realized that there are multiple processes that can lead to similar outcomes, and therefore that you cannot simply see the patterns occurring in nature and making inferences directly. Instead, what is needed is a model-based approach where you actually get to model the different processes to see which one fits better at the data you are observing. Bottom line, uncovering the history of a species is quite challenging. And so my work fits in trying to tackle this challenge by developing new methods that allow for more realistic inferences. Specifically, what I've been dedicating a lot of my time is to coming up with new methods that allow to integrate geographic and ecological processes into phylogeography. And I like to call this new integrative phylogeography, this new field. And today, instead of talking about all the different methods that I've developed over the years, I thought it was going to be better for me to illustrate their use in a particular system. Specifically, I'm going to be talking about, as I mentioned before, the impact of glacial cycles on generalist species in tropical mountains. And so what I'm presented in this slide is this figure of what is traditionally considered the role of Pleistocene cycles on differentiation of a species. So what you see here is the distribution of a species in green. In blue is sort of the glaciated areas. And you have two images. The one on top refers to an interglacial periods where the glaciers retreat towards the highlands. And then at the bottom, a glacial periods where you see an extension of the glaciers downhill. And so the idea here is that during these two different periods, populations have different levels of connectivity, such that during interglacial, populations become isolated because of their restricted distribution. Whereas during glacial periods, you have periods of connectivity in refugial distribution. Over time, during interglacial, populations are expected to accumulate differences, and given enough time, they may even lead to reproductive isolation, so to the origination of new species. This cycle of glaciation and interglacials is expected to have repeated multiple times, and therefore to have given rise to several different species. This is what it's commonly known as the species pump model, which each cycle of glacial interglacial promoting new species. Now, what is key to understand in relation to this model is the fact that it relies on species showing environmental specialization across elevation, such that during periods of interglacial, they remain isolated from one another. So this species pump model is expected to be particularly prevalent in tropical mountains, and that is because of something that Jansen proposed in 1967. So what you see on the slide right now is the environmental profile of localities at low, at high elevations during an entire year, both in the temperate zone at the bottom and in the tropical zone at the top. And because of seasons in the temperate zone, you see this overlap in the environmental conditions that a particular species experience in both environments, either in the low altitude or high altitude. And in contrast, in the tropical areas, because of the lack of seasonality, low altitude species and high altitude species do not show this overlap in environmental preferences, which allow them to become specialized. This ultimately is expected to lead, as I was saying, to segregation or isolation between species. So you can see the mountains on the right, where high and low elevation species will not share genes with one another and allow to become more differentiated. And in fact, there is quite a bit of evidence in support of this idea of environmental and altitudinal specialization in species. So you can see here a study by Cadena and Cespedes in 2019 that showed that elevational ranges in these birds actually are constrained, so that you see these transitions that different species occur at different elevations. And at an intraspecific level, even, you see evidence of genetic differentiation across elevation. So what you see there is on the y-axis, that FST represents genetic differentiation. And on the x-axis, you see elevation disparity or differences in elevation. And basically, you see this pattern where the farther apart in elevation populations are, the more genetically differentiated there are, suggesting again that they remain isolated during periods of interglacials. However, there are exceptions. Not all species have these narrow altitudinal ranges. One of those species is Akodon mollis. This is a generalist species that occupies Ecuador and northern Peru. That's its distributional area. And what is particularly striking about this species is the fairly large altitudinal range that it has, going from about 500 meters all the way to 4,500 meters. And of course, associated with that large range of elevation, there is quite a significant gradient of both biotic and abiotic conditions. So in terms of the biotic conditions, here I'm presenting some examples of the kind of environments where you can find these species, going all the way at the bottoms from a subtropical forest to a highland environment, grassland at high elevations. And as I was saying, associated with these elevational gradient, there is also significant changes in several environmental variables. Some of them are listed here, such as temperature, precipitation, UV radiation, and oxygen levels. You probably have heard a lot of examples of adaptation to hypoxia at high elevation. So the question is, how is this species able to tolerate all that range of environments? Also important in relation to the system is the fact that it has been strongly impacted by Pleistocene glaciation. So what I'm showing here is a map showing the infer depression in the snow line in the area where Akodon mollis lives. And you can see there are several places indicated by the red arrows where the snow line decreased about 1 kilometer. So it went down about 1 kilometer during glacial periods, which will have strongly impacted the areas available for Akodon mollis to live. And further evidence of these changes come from palynological studies that show a dramatic transition in the prevalence of species associated with this paramo environment, more of this grassland that I showed before, in comparison with subparamo environment. And as you can see, there is this nice transition where paramo used to be more prevalent, and now during the interglacial, it become less so. So this species showed at least the potential for adaptation, that there are several opportunities to specialize across that large environmental gradient. And it also showed the impact of glaciation. So the question is, how well does this species pump model that I presented before fit generalist species such as Akodon mollis? To answer this question, I decided to follow to divide it into multiple sub-questions. And so what I did is assess whether Akodon mollis shows population structure patterns that are consistent with a model of interglacial isolation, whether there is evidence of climate-induced distributional shifts, and whether demographic model of cyclical connectivity do in fact better explain genomic patterns in these species. So let's start with the first one. So here I'm looking at degrees of population structure during the period of interglacial, which is the one we're currently living. What I'm showing here is an haplotype network, and to orient you, basically what this graph shows is the size of the circle represents the number of samples in a particular locality. The different colors represent different haplotypes, and the distances between them, although you can see too many in this haplotype network, represents how different those haplotypes are from one another, which is expected to be correlated with the degree of isolation between populations. And just so that you can compare, here I'm presenting an example that I borrowed from a different study. What I want to highlight here is these long lines, which again represent the genetic distances between the samples. And so contrary to that example, you can see that in Akodon mollis, really there are not that many differences between different populations, suggesting again that they are not isolated from one another. To further show these results, I included here an analysis, this time not using a single gene, but a whole set of nuclear genes that I derived from a ddRat library. And what you see here is what it's called an admixture plot, in which each individual is represented with a different bar, and the colors represent the ancestral group to which these individuals pertain. And again, to help you orient to what we're looking here at, I present another examples. Typically, what you see when there is population structure is these bars of a single color, showing that each one comes from a different lineages, and there is no admixture between them. Whereas in the case of Akodon mollis, you see that there is some level of separation that is related to latitude, but overall, there is high levels of admixture across populations. I look further into this pattern by looking at a comparison between genetic differences on the y-axis and elevational difference. So this is plot similar to the one that I showed before during the introduction. And what you see is that in the case of Akodon mollis, we don't have a significant association between differences in elevation, so where different populations occur along the elevational gradient, and genetic differentiation, suggesting again that these populations are not isolated. So together, these analyses show that there is no significant population structure in these species. Moving into the second part, here I'm looking at the evidence for climate-induced distributional shifts. So do we see this pattern of range expansion and contraction associated with different climatic cycles? So to look into this question, I created ecological niche models by combining records of these species with environmental data and identifying the areas that are suitable to the species. And then I use this information to retroproject what the habitat suitability would have been back in the past, all the way to 120,000 years ago. And what you can see by this animation is that there are repeated cycles of contractions and expansion of the ranges. To further corroborate these inferences, I use a method that I developed in collaboration with Mike Hickerson. And this is a method called the geographic spectrum of shared alleles that allows you to make inferences about the origin of geographic expansions exclusively based on genetic data. So this is a nice way to contrast the method that uses ecological models, so it uses environmental data, with one that only uses genetic data. And what is nice in this particular case is that you see a nice correspondent in the location of where we predict the refugial area during glacial would have been. And that is represented by the black circle in both maps. Another thing that I want to highlight about this method that I developed is that contrary to other methods that use kriging to inform the possible location of refugial distribution, this method actually allows to use effective distances so that I can use not only 2D distances, latitude and longitude, but actually differences in elevation so that it's more reflective of the true distances between populations. This method also allows to infer how the geographic range proceeded. And again, there is a nice correspondence between the genetic data, what the genetic data suggests, and what the ecological niche model suggests in terms of habitat suitability and how the expansion proceeded. So in summary, for this part of the talk, we saw that there is evidence of climate-induced expansion and contraction cycles. So now that we have these two components, I wanted to test the last part of this puzzle. And this is whether we actually have evidence of population connectivity increasing in refugial area during glacial periods and population isolation occurring during interglacial periods, as represented by these circles separated by from one another and using different colors showing that they are becoming somewhat distinct. So the first way to tackle this question is was to actually look at changes in effective population time through effective population size through time. And what you see on this plot on the left is on the x-axis, you have time from the present to the past. So the present occurs closer to the y-axis and then moving towards the right, time and start to increase. And then on the y-axis, you have effective population size. The red line is the point estimate, and the gray lines are the confidence intervals around that point estimate. And what we see is what we will have predicted based exclusively on the ecological niche model, which is this contraction in the geographic range, which will lead to a decrease in population size. So we do see this decrease. And what is particularly nice about comparing these two methods is that the timing of those events also coincide. So there is this pattern where species went into refugial area during glacial periods. However, there is a little bit of a black box as to what happened after that period of time. So there is limited power in this analysis, at least based on the data that I use for it, in terms of inferring where there are cyclical changes, and it doesn't allow you to make inferences past that initial reduction in population size. Part of this is related to the fact that I have a relatively small number of SNPs in this analysis. So I now have a new data set with a much larger sample, both in terms of the number of SNPs, but also the geographic coverage of them. And so I'm rerunning this analysis and hopefully we'll have a better picture of it soon. But to actually look this question in more detail, I decided to go with demographic model. And what we do here is basically we simulate patterns of genetic variation based on particular models, and I presented two of them here, and then assess how well do these simulated data match the observed data, the ones that you collected from the field. And so just a few examples of the models that I presented here. On the left, you have an acyclical model in which there is this pattern of isolated populations during an interglacial that went back into a refugial population and stay in that configuration until all the genealogies coalesced. And of course, this seems to be a naive model based on what I showed before, but I think it was important to test. And I'm going to explain in a little bit what this model actually represents. I also tested the model where there is a single population throughout both glacial and interglacial periods, but I'm not showing that one on the screen. On the right, you have a model of the cyclical variation. So at the bottom, you will have an interglacial period right here. And then these populations merge into a single large population during glacial periods. And the text on top of these figures represent the different parameters of the models that I included. That is the effective population size during interglacial periods, the effective population size during glacial periods, as well as the time of when those events happen, with the idea of testing whether they coincide with both my ecological niche models and what is known about the glacial interglacial climatic variation. I included an additional set of models that allow for migration during interglacial periods. And this is important based on my previous results that show that there is actually no significant population structure in the population. And I'm not going to go over all the details. I run these models in FastSimCall for those of you that are familiar with this type of modeling. But bottom line, I found that the best supported model was the model that I expected, the one where there is differentiation into different populations during interglacials, and then they come in together during glacial periods. However, I have to say that there is relative modest support for an alternative model where there is now this cycling. And I think it has to do with the fact of how migration, how strong migration is during interglacial periods. So in the case where migration is really strong, basically the population will become effectively. So the data currently do not separate between these two models. Related to this is also how important the summary statistics that I use to infer these models are. So I'm currently now working on incorporating a set of summary statistics that I developed that include information about the geography, so where species occur on the landscape, that provide further information to better discern across these models. But what I think is important to see when looking at the parameters is that it again coincides with some of the other evidence that I gather on the system, and that is that interglacial population size was much bigger than glacial population size. So corroborating this idea of them coming together during periods of glacial cycles, and also a significant reduction in the number of individuals that there are. So taken together, I show you that there is no significant population structure in Akodon mollis, that there is evidence of climate-induced cycles of expansion of contraction, and also this preliminary support for uninterrupted GIF flow even during periods of interglacials. So coming back to the question that I posed as the leading question for this talk, is the fact whether the species pump model fit generally the species such as Akodon mollis, and the answer is no. I think this is a poor model for this species. And you can see that in the little scheme on the left, where what I'm showing is what I think is more realistic for this species. And that is that even during periods of interglacial, because of this large altitudinal range and environmental range, populations are not completely isolated from one another. So you can see that also in this model that represents, again, the glacial interglacial changes. And what I'm showing as the main difference is that geoflow is not completely absent during periods of interglacial. So that makes the response of this species quite different. And it all has to do with its biology, how large its environmental tolerance is. So the take-home message for my talk is first that I think integrative phylogeography has a lot of power. And by integrating information about the biology, species, and the environmental setting of where they occur, you can get more accurate inferences about the species' evolutionary history. The second point that I think is important is that there is a general, that the expectation that there are a single model that fits all is probably incorrect, because in general, concordant responses to past climatic events will be rare. And if you ask ecologists, and I have talked about this with ecologists, they typically laugh at us when we think that a species responded all in a similar manner. And this has to do with this debate about Clemensonian responses to succession. So I think similar patterns occur in a longer time period with over time, even under the same climatic pressures, different species responded in a different manner. And finally, I think that species ecologies and geographic setting are quite important, and therefore they need to be incorporated in the analysis. But I do think that there may be some general responses, so that is the model that I found the most likely for Akodon mollis the same for other generalist species? We don't know yet. And I think the tools that are currently available allow us to ask this question by making comparisons both within the same system, so multiple species within an area, as well as across different systems that differ maybe in the environmental conditions as well as the species biology. And with that, I just want to thank all the people who helped me throughout the years in this project, as well as that's an error, that shouldn't say funding, that the resources, which is the computational resources that allow me to run this analysis, as well as the funding. And finally, shameless plug, I'm actively recruiting people in my lab. So if you are interested in this sort of question, feel free to contact me. I will be available in Discord later on. Thank you very much.
Thank you, Dr. Diego Alvaro-Serrano. This was great. By the way, please note that we are a bit past time, and on purpose, I did
Oops.
not let you know about that because your presentation was going great and I wanted you to focus on that. To people that have joined us, what we are doing now is, although we are a bit past time, we are going ahead and we are continuing with question and answers. If that's all right for you, Dr. Alvarado-Serrano, is that
Yes, certainly.
okay?
Yeah, we'd be happy to answer the
Awesome.
question.
And just a reminder to everyone else, the next talk would be by Annie Yoder. It's starting at 3:50, so it hasn't started yet. Okay, I ask people from UNC Charlotte to please hold with us while we go through the questions and answers. I will help Sophia with questions in English. You may also submit questions in Spanish, and then Sophia will manage those for us. Dr. Alvaro-Serrano, there is a question from Francisca Almeida. She says, can you please repeat the name of the program you used to test the alternative demographic models?
FastSimCol2.
Can you type it in the chat so she can have easy access to that? You may send text messages to panelists and attendees and everyone will get that.
Yes,
Fantastic.
that's not the only one. I can mention a couple more. And actually one that I like better is Slim 2, but that's the one I use for this project.
One suggestion would be, since Dr. Alvarado-Serrano is with us in our Discord server, maybe you would feel interested, people that are in this call with us, if you want to join us there, especially because later we are going to have our poster sessions there. Maybe you want to join us there and send messages to us. You may use the icebreaker room to send messages to the speaker and get answers there. Okay. Do we have any other? We have another question from Nuno Martins. Which sort of data can we input into FastSimCol?
So FastSimCol allows any kind of genetic data, maybe mitochondrial data, chloroplast data, it could be microsatellite, or it could be sort of genomic data, all these libraries. However, there is something to say as to the ability of the program to make inferences, and that has to do with the number of data. What you want to do is have the larger number of independent loci. So although the program allows you to use a single marker, you may end up with flat posteriors for pretty much all your parameters and models. So you may not be able to say much about it, but the program allows it.
That is a great short explanation. Thank you. I would like to know if anyone has any other questions in Portuguese or Spanish. I know that we are a bit past time, but maybe we can squeeze in more questions here. Sophia?
There is a question from Mariana Gilardi. She says, thank you for your talk, Dr. Diego. Did you plan to test these models that you have created with other species?
Yes, so I didn't have time to show. I'm actually in the process of doing it. I'm right now using a community phylogeography approach, which I'm using other rodents that coexist with Akodon mollis and that vary in their degree of environmental specialization. And so as to make comparisons. And we are in the process of extending this job to other Sky Island systems. So we are submitting a proposal to NSF to look into comparisons with other Sky Island systems where there may be differences in terms of the time of the origin of the different mountains and differences, ecological differences between the species as well.
I don't think I see other questions. Do you see any other questions right now, Sophia?
No, not more.
Okay, so since Dr. Diego Alvarado-Serrano is with us on Discord, if you want to, you may move there later, and I will create a chat room on the lobby icebreaker with his name, and I will call you there, Dr. Diego. So if anyone wants to interact with him, please use that channel that I'm creating now. Thank you, everyone.
That would be great. Thank you very much.
Thank you, the UNC Charlotte team, for putting this together. We are ready to close this. Dr. Serrano, this was fantastic. Alvaro, that was fantastic. Awesome. Thank you so much.
Sure. Thank you. And thank you for inviting me again.